What's good, class, and welcome back to another edition of Mr. Fitzgerald Biology class. Today we're starting to talk about things called macromolecules. There are 4 macromolecules: carbohydrates, lipids, proteins, and nucleic acids. And today we're going to focus on 2 of those, as carbohydrates and lipids. We've already kind of talked about lipids and touched on those with the cell membrane, so we're going to review parts of those and add a little bit to it. But we're going to start off with carbohydrates today. So as we dive in, some important things to know. Macromolecules, the prefix macro means big. So anytime you see macro there, that means something that's big, like macroeconomics is thinking about the big economy. But with us in biology, we talk about molecules, so bigger molecules, right? So here would be an example of a very big molecule. Each of the little dots that are on here would represent an atom, and you can see all these connections that are there and how folded this thing is. Very complicated, so a very large molecule overall. So there are 4 types, as I said before. Carbohydrates are the first type of macromolecules. We have lipids as well. Proteins, and this would be an example of a protein here, a very big structure, and then nucleic acids, which we're going to touch on last. Okay, so this is kind of the order in which we're going to present these as well, so it kind of keeps a running list for you. All right, so let's talk about carbohydrates. And when I talk about carbs or carbohydrates, I'm not just talking about food labels and what you see on like nutritional facts. What I'm going to be talking about is sugar. So sugar is a very broad category. There are things that are very simple sugars, there are things that are very complicated sugars. And it's all based on the amount of molecules that are together. So carbohydrates are a great source of energy. You crave sugar because your body can get the most amount of energy with the least amount of work from it. You can use fats as a source of energy as well. It's a little bit more of a complicated process. So your cells are set up to use carbohydrates as a fast source of sugar. These compounds typically have a lot of carbons and hydrogens. You'll find that as a theme in all of these things because all these macromolecules are called organic compounds. And what organic compounds need is a lot of carbon as well as a lot of hydrogen to be able to get all the components together properly. Carbon is the best atom for this because it can make 4 different bonds simultaneously, so it can connect to so many things. And hydrogen is just so abundant, it usually fills any gaps that we need. Also, oxygen is involved in here, so we usually get this kind of standardized formula of C2O with some number here. Okay, so for an example, if I put the number 6 here, what I'm going to do is multiply 6 all the way through. So I can get some more complicated sugars by just kind of using the same pattern, because it's always double the number of hydrogens as there is carbons and oxygens. And the number of carbons and oxygens are always the same. So if I have a 6-carbon sugar, right, if I put the number 6 here, when I multiply that 6 all the way through the parentheses, I'm going to actually get C6H12O6. Now, hopefully that formula looks a little familiar to you because that is the chemical formula for glucose, or the sugar that most of you are craving right now if you want a snack. All right. So we'll talk a little bit more about that. There's glucose there on the side, labeled, showing you how it actually structures itself when we get all those bonds together. But notice all of these different sugars. I have lactose, a common sugar found in milk. This is a structure called chitin, which is in your nails and hair, and also makes up a cell wall for fungus, which is kind of interesting. But all of these share one thing in common. They all have hexagons. So when you're trying to identify what type of macromolecule something is, which you will have to do on tests and quizzes, you're going to have to look for the key shape. And for sugars or carbohydrates, it's hexagons. That's an easy giveaway for them. All right, so always remember that carbohydrates or glucose, which is the most common one we speak of in this class, are going to be represented by hexagons. Okay, so let's kind of continue on that train with some carbohydrates. Now, there are simple carbohydrates like glucose. Okay, it's arranged in one hexagon kind of ring, and these are called monosaccharides. Okay, there are simple sugars. We've already seen with cellulose, which is a more complicated sugar, that the suffix saccharide here means sugar, and mono means one. So these are single sugars, monosaccharides. Okay, monosaccharides mean single sugars, and these are the ones that are easiest to break down. So they're not stored up energy. It's energy that we can quickly access and go for. Okay, so just another reminder that saccharide is just a fancy way to say sugar. So the next time you're at the table and you want some sugar for whatever you're eating or drinking, ask somebody to pass the saccharides. They might look at you crazy, but it's a way to practice the vocabulary, and through practice you start to get it a lot more. Okay, so Looking at something with 2 sugars, well, if a monosaccharide is 1 sugar, 2 sugars should be something like double. So a lot of students like to think it's a bisaccharide, thinking like bicycle, 2. In this case, we don't use bi in science classes. We use the prefix di, meaning 2. Okay, so 2 monosaccharides will be together to make a disaccharide. So mono, remember, means 1, and di means 2. So if you see 2 hexagons together, that would be a disaccharide, or a 2 sugar. Okay. Now, we have polysaccharides, which we've already kind of seen with the cell wall and cellulose. Okay, so polysaccharides are when you have many monosaccharides together. Sometimes in single chains, sometimes they make long networks like this starch category over here. And then this one over here, glycogen, this is how you store up sugar in your muscle tissue. So when you have extra sugar molecules that are available, you store it up in this very complex polysaccharide called glycogen that you can then break down and release the individual sugars so you can break those down for energy as well. Okay, remember, poly means many, saccharides means sugar, so it's polysaccharide, there's many sugar. All right, so now we're on to lipids. Now, lipids we've already sort of discussed before. You'll notice this slide looked kind of familiar because I borrowed it from what we've already done. Why reinvent the wheel? So in here we have lipids are mostly a lot of carbons and hydrogens. We've already spoke about that. They're these long chains of carbons and hydrogens. Typical examples are fats, oils, and waxes. So I have a bunch of lipids over here that you can kind of remember that process there. And the big idea of them is they also store energy. And the reason they store energy is they have these long chains, meaning multiple chemical bonds holding these carbons and hydrogens together. Therefore, I can break all those bonds to release energy, just like I can break up sugar molecules bonds to release energy. This is a little bit more of a complicated process for the cell, so it doesn't do it first. It uses sugar, which is easier to break apart first, and if it needs to, then it starts to break down fat. All right, so some examples of lipids. So showing you all those lots of carbons and hydrogens here and all these individual lines. Those represent chemical bonds. So that is energy that is stored up in that molecule. Now, just like how sugars were broken down as like a monosaccharide, a single sugar, and polysaccharide as many sugars, lipids are kind of broken down similar, but it's a little bit different. We have fatty acids, which are kind of the single units of lipids. Okay, so if I get a bunch of fatty acids together, I have a lipid of some sort. So the easiest way to think of this is fatty acids are just large chains of fatty things. Okay, so lots of carbons and hydrogens. They usually have this head group so we can react and put them together in pieces. Sometimes they get attached to different groups to make more complicated things, but at the base they are just like this. Okay. They're the building blocks of lipids. Like I said, we can make more complicated things the more fatty acids we have, just like we can make polysaccharides with the more monosaccharides we have. So it's kind of the same kind of process here. A lot of things build. So we can have what are called saturated fats. So fatty acids can come in 2 varieties. There are saturated fats, which are long chains where every single carbon has 4 individual bonds to it. So if you look at it, it's covered in hydrogens. Okay, so hydrogen all the way through. Every little space where there's hydrogen is full. The word saturated means to be full. Just like if you got rained on at some point, sometimes you say your clothes are soaked. Well, I could actually say your clothes are saturated with water because they're completely filled with water. There's no more space. It starts to drip out. So in this case, right, I can't fit any more hydrogens here. I'm full. There's no more that can fit, so I'm saturated with hydrogens, okay? On the other hand, there's what's called unsaturated fats. And this is where you have gaps. So if you're looking across a fatty acid here, you see that it's got all these carbons and hydrogens, but there's one spot where there's a double bond, okay? And just because of that, there's a gap. And so now it's unsaturated. It's not fully filled with hydrogens. There's a little space where I can actually fit some more. In doing so, I would lose the double bond, but I would actually have a full set of hydrogens all around the thing. I'm trying to think of the word there. So that's the difference between saturated fats and unsaturated fats. Unsaturated fats have a little gap where there's no hydrogens present, and saturated fats have no gaps. They're completely full. They can't fit any more hydrogens on them. Okay, so comparing between the two. Saturated, full of hydrogens, nowhere else to go. I can't fit another one in there even if I wanted to. In the unsaturated fats, I do have a little gap that's formed here. So this is called a cis saturated fat because it's kind of bending it. You've heard of trans fats. That's where actually this piece would be up above, so it makes kind of like a staircase. So trans and cis fatty acids are a little bit different when they're unsaturated, but you don't have to worry about that for this class, only if you're really worried about dieting. All right, so that's the difference between cis and trans, or sorry, it's not cis and trans, that's for diets, from saturated and unsaturated fats. Now, one of the last ones I wanted to touch on, just to remind you again, is phospholipids. That is a very specific type of lipid that's predominantly used in the cell membrane. So, it's the area that has that hydrophilic head, or that head that likes water, as well as two hydrophobic tails, or the areas that hate water. And remember, because they have these non-polar hydrophobic tails, the heads will arrange themselves on the outsides, so it makes a bilayer where the two tails are actually interacting with each other from both of the phospholipids, and then the two heads will be interacting with the water inside and outside the cell. So it's a major component of our cell membrane. It's important in that selective permeability for the cell membrane, and it keeps the cell really as a cell, otherwise we wouldn't have it overall. That's all I have for today with the macromolecules of carbohydrates and lipids. We'll talk more about the other ones in the future. Have a good one.